Good afternoon, everyone. My name is Jim Gardner. I'm one of the curators here at the Royal Tour Museum, and I'd like to welcome everyone to our one and only online presentation in this year's Royal Tour Museum Speaker Series, which is sponsored by our Museum's Friends Society, the Royal Tour Museum Cooperating Society, who fund a lot of extra things around here, including the speaker series, research visits, some of our exhibit work, and so forth. So we're, as always, quite grateful for their support. Today, we're going to be talking about a somewhat unusual topic, the fossil record of parasites. I have to confess, it's not something I felt or I thought very much about until I stumbled across our speaker's recent review paper and reached out to him to see if they would be willing to give a talk. So we're quite happy to have them. So our speaker today is Tommy Leung. He's a lecturer in parasitology and evolutionary biology at the University of England in Australia. Their main research interest is on various aspects of parasitism, symbiosis, including the evolutionary ecology of parasites, macroecology of parasitism, and host characteristics and environmental variables that shape parasite communities. They also write the interesting Parasite of the Day blog, which is updated monthly, I believe. And that's always makes for some interesting reading and got some riveting photos as well. So I encourage you to check that out. So that's my introduction. I'm a bit out of practice for these online ones. So I'd like to welcome Tommy and take it away, Tommy.
All right. Thank you very much, Jim. So first of all, I want to thank, of course, Jim Gardner for inviting me to this speaker series and, of course, the Royal Taro Museum as well. This is not exactly a position that I ever imagined that I would be in, given that I'm a parasitologist and most people who give talks for this speaker series are paleontologists. So it's a great honor for y'all to invite me to talk about the topic that I'm going to be talking about, which are fossil parasites. So I'm just going to share my screen here so that the presentation is underway. All right. Great. So let's get started. Okay, so for this talk, I'm going to be talking about essentially parasites in fossil record. As Jim mentioned, not something that most people usually think about when it comes to fossil things. Most people probably think about dinosaurs and mostly vertebrate organisms, and certainly not things like parasites. So I'm going to be giving a bit of an overview about what are found in the fossil record in regards to parasites and the kind of insights they can offer us about basically the history of life on Earth. So I'm going to start by talking about something that is really different from, I guess, most of your speaker. I'm going to be talking about parasites in the here and now, because this would provide us with some context as to parasites in fossil record. So parasites are essentially ubiquitous. They're found in every single habitat. Basically, everywhere you find living things, you will find parasites, including parasites themselves also have parasites infecting them. Even in some very extreme habitat, you'll find parasites living in the organisms that live in those extreme habitats. So for example, there are deep sea amphipods, low crustaceans that live in deep sea trenches, over 7,000 to 10,000 meters below sea level. Yet in this kind of crushing environment, dark, oppressive darkness, you have parasitic roundworms, nematodes that live inside the body of these amphipods, and as you can see, thrive inside of them. In this photo, you can even see the coils of a nematode coiled up inside the body of this amphipod. So even in places like that, there is no escape from parasites. Additionally, parasites have also embedded themselves in many forms of ecological interactions. Indeed, they depend upon these ecological interactions as a mean of transferring from one host to another. Many parasites rely upon trophic transmission that are based around predator-prey interaction. That's just a fancy way of saying when one animal eats another animal. So many parasites have their larval stage inside prey animals, which are then transferred into a predatory animal when that predatory animal eats them. So whenever you watch like a documentary where a dolphin's eating a fish, or a great white shark eats a sea lion, or a lion eats a zebra, you are also implicitly witnessing parasites being transmitted from one host animal to another. Now, in addition to being embedded in various different kinds of ecological interactions, some parasites actually play keystone ecosystem roles that can influence and shape the ecosystem through the actions they have on the host. So for example, on the coast of New Zealand, there is a very common bivalve called the New Zealand cockle, which are found on sand flats and mud flats. They're so common that if you walk along the coastline in some of these areas, the mud flats is completely covered in these bivalves. Now, most of the time, these bivalves are burrowed underneath the sand. However, there is a little fluke that actually infect these bivalves and burrow their way into the foot. And as more and more of this parasite accumulate in the foot of a cockle, they lose the ability to burrow themselves back into sand, which leaves them exposed to the predators like oyster catchers and seagulls that happens to be the final host in the parasite's life cycle. Now, there is an unintended consequences of this is that in areas where this parasite is very common, there are many cockles that are basically left stranded on the surface of the sand. But this provides a habitat for a wide variety of different animals that rely upon some kind of hard substrate. So for example, limpets and barnacles and many other organisms can now settle on these otherwise uninhabitable mudflats. In fact, when scientists compare areas where you have high infection prevalence of this particular parasite in cockles versus areas where the parasite is comparatively rare or even absent, they found that in areas where cockles are heavily infected, there is also greater level of biodiversity as well, because the parasites through the action are essentially creating free real estate for a variety of other small organisms. So many parasites have key ecosystem roles that are unseen because they're hidden inside the body of the hosts. In addition to being ubiquitous across different ecosystems, they're also ubiquitous across different phylogenetic groups as well. In the animals alone, parasitism has independently evolved at least 100 times, and you'll find parasitic representatives across many different groups of animals, even in groups of animals you wouldn't normally think of as being parasitic. So for example, in the mollusk, there is actually over 5,000 species of parasitic snails and bivalves. So snails, normally something that people, if they do think of in terms of parasites as being host to parasites, but there are snails out there, and many of them, that actually live a parasitic lifestyle. Furthermore, there are also phyla where most of the known species are actually parasitic. So for example, in the flatworms, there are of course many species of free-living flatworms, but the majority of flatworms on this planet come in the form of flukes and tapeworms. They compose of about 90% of the entire phylum of flatworm. It seems that evolving to become parasitic is the best possible thing to have happened to the evolution of this particular group of animal. And then there are things like nematodes, roundworms. Nematodes are also ubiquitous in the environment. They live in all kinds of habitats that most other self-respecting animals do not live in. However, parasitism has evolved at least 15 times among nematodes. So on 15 different occasions, different lineages of nematode go, you know what's good? Living inside of another thing. So parasitism is extremely common lifestyle. And this is just the animals. Let's not get started on, say, the plants, which parasitism has independently evolved multiple times within the flowering plants, and then the fungi, which are extremely good at parasitism, and all these kinds of single cell organisms out there, which are also parasitic and causes major infectious disease. So parasites are basically found everywhere on the planet. You can't get away from them. What about in the fossil record? Well, in contrast to that, fossils of parasites are actually extremely rare, simply because many parasites have body plans and body sizes that are not very conducive to the fossilization process. Think of something like, tapeworms that grow to multiple meters long, as big as they are. They're very soft, squishy animals, and soft, squishy things don't tend to get preserved in the fossil record. Furthermore, I guess this is not something that paleontologists usually considered or has considered in the past. Parasites, they usually think of their study organism and not necessarily the things that live on those said organism. However, this has changed over recent years as there's growing awareness among paleontologists that, hey, you know what, parasites, they're kind of important. Furthermore, there's also improvement in various different imaging technologies. So for example, this particular spectacular finding comes from fossilized fly pupa that were found in France dating back to the Eocene period, around 34 to 40 million years ago. And thanks to the use of X-ray microtomography, we're able to see that there are this, parasitoid fly, a developing parasitoid situated within the inside of this fly pupa. So in the past, if you look at this fly pupa, it just looked like an ordinary fly pupa. But whereas you subject it to X-ray microtomography, you discover that, oh wow, there is like a developing parasitoid found inside of it. So why should paleontologists even care about parasites in the first place? Well, as I've established, parasites is an integral part of biodiversity in ecosystems on this planet. To ignore parasites is like reading the story of Book of Life and skipping half the chapters, essentially. Furthermore, understanding parasites can also help understand their host, because parasites can provide some very key insight into the livelihood or the lifestyle of the host, as well as the environment that they find in, as I'll explain a little bit later on. So given that this particular talk, I'm limited to an hour, I'm just going to provide a bit of an overview of parasites in the fossil record, because if I were to give an exhaustive overview, that would literally take hours. And as much as I would like to talk about this topic for literally hours, I don't think our audience have that level of patience for this. So I'm going to be providing you with the highlight. You can kind of think of this as the sample platter of parasites in the fossil record. So I'm going to be focusing mostly on animal parasites, and given we're talking about animal parasites, let's start the story in the Cambrian period, where I guess most of the animal initially evolved. So there are these worms called prepulid worms that lived during the Cambrian period, and there were some recent findings of these kind of little bowling pin organisms that are stuck to its body. It's not really clear about what kind of symbiotic relationship occurred between these little bowling pin things, for the lack of a better term, and the host worm. But they're found on multiple different species of these worms, and they're just kind of stuck to the body. It doesn't seem like they actually penetrate through into the body, so it could be kind of a commensalistic relationship. Either way, it's probably not great for these worms that are mostly burrowers to have these things stuck onto them, because they kind of get in the way. So the exact relationship between these bowling pin organisms and the host worm is not entirely clear. There are also other fossil parasites that's been found from Cambrian deposit. One most well-known example are these larvae of pentastomids, or better known as tongueworms. So pentastomid is a rather enigmatic group of animal that wasn't until relatively recently we were even sure what they actually are, simply because, look at them. The adult stage looks like worms, and the adult stage of pentastomids that are found today are actually found in the lungs of vertebrate animal, mostly in reptiles, and they look like worms. They don't look anything like, an arthropod or anything like that. It wasn't until approximately about 20 years ago that, through the combination of analyzing and comparing the DNA as well as comparative anatomy, that researchers were able to figure out that, these things are actually crustaceans. They don't look anything like what you would expect a crustacean to look like, and certainly not for them to be living inside the lungs of land-dwelling vertebrate animals, but they are crustaceans. They are an extremely derived group of crustaceans. So this in turn just raises even more questions about those Cambrian pentastomids, because basically the question is, what on earth were they doing back in the Cambrian period? Because the kind of animal that they live in, or the habitat that they live in, or the adult stage live in, are not set to evolve for at least another 200 or 100 million years at least. And so there was kind of a period between, say, the Carboniferous and also the Cambrian period where it's not clear that, okay, if there are fossils of larval stage of these peculiar organisms dating back to the Cambrian period, what were they getting up to in the meantime before they become parasites, internal parasites of land-dwelling vertebrate animals? There is a fossil that was found from the Silurian period, which might give us some insight into what they might have been getting up to. So in this particular fossil, the researchers describe what appears to be a pentastomid adult attached onto the carapace of these little ostracods, these little clam shrimp-like small crustaceans. So this might provide us with a clue as to the evolution of this particular group, that before they became internal parasites living in the lungs of land-dwelling vertebrate animals, they were actually external parasites of crustaceans, maybe? I know that there are some researchers who are a little bit skeptical about this finding, whether this thing attached onto this ostracod is actually a pentastomid. But even if it is, it just raises more questions because, like, for example, how did they make the jump from being ectoparasites of crustaceans to becoming internal parasites of vertebrate animals living inside the lungs? How did that transfer occur? So this particular group of animal is just full of enigma that are still yet to be answered. Now, there is also other partial remains of parasites that's been found in fossil records. So, for example, there is an assemblage of fish dating from the late Devonian period that's been found to have these little hooks on them. So the fish in questions are things like placoderms, which are the armored fishes that were very common during the Devonian period, as well as the Echinoderms, which are a group of extinct fish called the spiny ray sharks. Both of these groups of fish are completely extinct now. They went extinct at the end of the Devonian period. And so some of these specimens have these little circles of hooks, mostly found on their gills, as well as on their abdomen and on their fins. And for a while, it's not really sure what they actually are. They suspect that, paleontologists suspect that they might be some kind of parasites. But when I saw them, I realized that the thing they remind me the most of are actually monogeneans. So monogeneans is a very diverse group of parasitic flatworms. There are thousands of known species, and there's probably a lot more which are undiscovered. They have this specialized organ called an opus haptor, with which they attach themselves onto their fish hosts. They're found on fish just basically all over the world in every single ocean, as well as freshwater habitats as well. They use this opus haptor to attach themselves onto the skin or the fins, or in some cases the gills of fish, pretty much exactly as described for these little circlet of hooks that are found on these Devonian placoderms and Echinoderms. So it's quite likely that this circle of hooks actually came from an animal that looks something like that. And it's actually amazing to think that these guys are still infecting fish today, because if they were found back in the Devonian period, that means that they would have gone through multiple mass extinction event, and every single time they were able to bounce back to infect new groups of fishes as they have evolved. This is like a really inspirational story, I think, with these Devonian monogeneans, if they are monogeneans. Now, fish are also infected by other kinds of ectoparasites as well. So there are what appears to be parasitic copropods that has been found in the gill chambers of some early Cretaceous fish dating from about 120 million years ago. These particular crustacean rather resembles the parasitic copropods that are found on the gills of fish today. What many people might not be aware of in regards to copepod is that most people, if they have heard of copepods, they usually think of them as tiny little zooplankton organisms floating in the sea. However, about 1/3 of all known parasitic copepod are in fact parasitic, or symbiotic at least, living inside the body of other marine animals. And they are, these little monsters are amazing. And I call them little monsters because if you look up pictures of them, you will completely reevaluate anything that you have, any assumptions you have about what a crustacean to look like. It's like as if, these parasitic copepod had a look at the playbook of what a crustacean is supposed to look like and just tossed it out the window and evolved into all kinds of extremely bizarre form. So there's some evidence to indicate that they have been parasitizing fish since at least the early Cretaceous period, if not earlier than that. And speaking of earlier than that, there is also another group of common parasitic crustacean found in fish, which are parasitic isopods. So isopod is the order of crustacean that things like slaters belongs to, slaters and pillbugs. Some people might have heard of Baphonomus, which are those really interesting looking giant isopods from the deep sea, but there are also parasitic representatives as well. So there are fossils of what appears to be parasitic isopods dating back from the Jurassic period, about 168 million years ago. They have claws that seem well developed and suitable for clinging onto the body of fish, as well as mouthparts that are piercing mouthpart that seems to be for stabbing into fish and sucking out blood and other kind of bodily fluids, unlike most isopods, which mostly have like slicing or chewing mouthpart. Now, parasitic isopods on fish are very common today. One of the most infamous example is probably the tongue biter louse, arguably one of the most famous of all isopods because it was actually featured in an episode of the Colbert Report. But the tongue biter louse, or should I say tongue biter lice, because there's actually multiple different species of them infecting different fish, are just one out of many different types of parasitic isopod. They mostly belong in this particular group called the cymophoids, and they are found on all kinds of different fish body parts. Some of them live in the gills instead of the tongue. Some of them are actually clinging to the surface of the fish, often on the head, that I like to call face huggers. And there are even some that actually burrow their way into the abdomen of the fish. That's the reason why I find it a little bit quaint when people freak out about the tongue biter parasite, because it's like, whew, you have no idea what else these guys get up to. Now, while all the examples I've talked about is where you have parasites themselves being preserved as fossils, unfortunately, that's not very common, for the reasons I talked about previously. Many parasites are kind of small and they're squishy, so not really good material for fossilization. However, there are some parasites that actually leave behind traces, signature marks on their hosts that are permanent and are very, very distinct. So for this example, I'm going to be talking about the Boperids, which are another group of parasitic isopods. But instead of being parasites of fish, they're actually parasites of crustaceans, specifically crustaceans in the decapod order. So this is the group that includes things like crabs and shrimps. And when they get inside of the crustacean host, they move into the branchial chamber, which is where the gills are. And their presence actually induces this characteristic bulge that you can find on the carapace of these infected crustaceans. Indeed, there are fossils of decapods that date back from the early Cretaceous period or even to the late Jurassic period that have these characteristic bulge. Interestingly enough, the late Jurassic is also a period when many of the recognizable crabs started evolving as well. So it seems like as soon as the crabs arrive on the scene, there are these parasitic isopods that start infecting them as well. Now, this is how these isopods actually look like. They kind of like if you get a pill bug and run it over with a steamroller in a kind of a cartoon, like Looney Tune cartoon kind of a way. And you can see on the photo on the left, how they're actually situated inside the body of the host. So this particular shrimp has a transparent exoskeleton, so you can see through it, see the little isopod. They have a mating arrangement that is similar to that of deep sea anglerfish, where you have like a really large female and what's known as a dwarf male attached to the female. So you can see here, even in the photo, there's this tiny little male isopod located between the pleopod of the bigger female isopod. So Boperid isopods are not the only parasite to leave behind characteristic marks or traces on their hosts. If you look at bivalve shells, both fossils and modern, you'll find that some of them have these little pits on the inner surface of the shell. Some of them even have these what's known as like igloo-like structure that are located on the inner surface of the shells. These particular structures are actually pearl formation, because while most people think of pearls as being like jewelry, these things are actually like pearl formation is actually an integral part of a bivalve's or mollusk immune system. When you have intruders coming in, like some kind of parasite, oftentimes the bivalve would respond by secreting these pearlescent substance, which essentially entune the parasite alive. And there is one particular species, or well, actually one particular family of parasitic flukes that are mostly associated with these kind of pearl formation. And that family is called the Gymnophalidae. So you can see examples of that there. On the top left, you have a mussel that's been infected by these Gymnophalidae in the process of forming some pearls. And right underneath that is one of these flukes. So each of these little pearls, little natural pearls, have actually entombed inside of them a dead little parasite. In fact, when scientists cut through some of these pearls, they were able to extract DNA and other kind of remnant remains of deceased parasites inside of them. So essentially, the bivalves respond to the presence of the pearls, respond to the presence of these parasites by entombing them alive in this pearlescent material. Now, pearls have been dated back all the way. The earliest known example are from the Triassic period. There are some pearls that's been known from bivalves dating from the Silurian period over 400 million years ago, but those pearls, given that there is like a 200 million year gap between those pearls and the earliest known pearl that is recognizable as modern pearls, it's quite likely that whatever causes the pearls in the Silurian bivalves might be a little bit different to what causes pearls in the Triassic and Mesozoic bivalves, and as well as modern bivalves as well, which are this particular family of fluke. Now, pearls have also been found in other types of fossil mollusk, and one of them is the ammonoids. So ammonoids are an extinct group of shelled cephalopods, so relatives of squid and octopus. These guys used to be extremely common, especially during the Mesozoic era, around about the same time when, like, you know, the non-avian dinosaur, the recognizable megafauna for everyone. They used to be as common as fish in the ocean, and they range in size from tiny little things that could fit in the palm of your hand, all the way up to some that are about the size of like mining truck tires. Now, some of the shells of these ammonoids have also been found to have these little pearl pits on the surface of the shell. And even though you'll be tempted to think that, like, oh, is it those flukes that causes them as well? The patterns of pitting on the ammonite shell, as well as where they're actually found on the shell, which is on the outside, indicates that there's probably some other kind of causes for it. There's some other parasite possibly that are causing this pearl that is different from what is causing pearls in, say, bivalves. So a little bit of unsolved mystery here. So something is causing like pearls on these ammonites, but we don't know what exactly they actually are. Now, there's also other kinds of hard-bodied organisms that have traces of their encounters with parasites. So an example of that is the Echinoderm. So Echinoderms is a group of animal that contains the likes of sea stars and sea urchins and crinoids. This particular group of animal has actually an endoskeleton. So it looks like a shell, but it's actually located on the outside, on the inside. So technically it's an endoskeleton. But because of this, they also preserve records of them being attacked by parasites. And in this case, the parasites that are attacking them are, as foreshadowed by before, parasitic snails. So during the Paleozoic era, between 500 to about 250 million years ago, there was a family of snails called the Platyceratidae, which are parasitic on various different kinds of echinoderms, mostly on crinoids. And many of them drill into the stalks of crinoid or the main body, the test of the crinoids. And when they do drill into them, they leave behind these characteristic mark of their attacks. And because in this case, the parasite themselves, unlike most of the other parasites I talked about, actually have a hard body part in the forms of the shell, some of them are preserved in situ with their host as well. So this is like an amazing kind of fossil parasite host system to study, because oftentimes you'll find like well-preserved record of the parasite located on the host. Now, the Platyceratidae died out at the end of the Permian period, about 252 million years ago, because there was this thing that happened called the end Permian mass extinction event that essentially wiped out over 95% of everything that was alive. It was like a really, really big deal, so much so that it's been called the Great Dying. So the Platyceratidae, as well as the many echinoderms that were alive at the time, they went extinct. Echinoderms used to be really, really abundant during the Paleozoic era. The crinoids and an extinct group called blastoid used to form these like massive undersea forests, which would have been a great time for these Platyceratidae snails. However, the end Permian mass extinction put a stop to all of that. And so the echinoderms would survive, but the Platyceratidae did not. However, the Platyceratidae had a spiritual successor in the form of a new family of snails called the Eulimidae. So the Eulimidae is yet another family of snails that have independently evolved to be parasitic on echinoderms, and fossils as well as the traces of them have been found on echinoderms dating from the Cretaceous period, around about 90 or so million years ago. And they leave behind these very characteristic drill marks on the body of the host. Now, how do we know that these particular drill marks are left behind by Eulimidae? Well, the reason is that we actually have modern examples to compare them to. Eulimidae are still parasitizing echinoderms today. They are most of the 5,000 or so species that I mentioned in passing earlier. And they come in many wonderful and bizarre forms. You have some species of Eulimidae that actually drill into the spines of sea urchins, and they induce the sea urchin to grow this gall that forms a habitat for it. It's kind of rather comparable to the likes of like gall wasp and other things that form galls on leaves as well as branches of plants. There's also other kinds of Eulimidae that, for example, this species here that live in the cloaca of sea cucumbers. And they're not the only one to do it as well. There's actually a wide variety of animals that make their home inside specifically the cloaca of sea cucumber. For some unknown reason, there seems to be an invisible free real estate sign floating above of sea cucumbers in the sea. And that's not even the most bizarre of the Eulimidae, because this is probably among the most bizarre. This is a snail. Once again, this is a snail. Even though it doesn't look anything like a snail, it lives as an internal parasite of sea stars. And short of comparing its DNA, the only way for you to figure out that this is actually a snail is by looking at its villiger or larval stages. You can see that they look like tiny little shell, they have shells and they look like essentially miniature snails. But once they get inside the body of a sea star, all bets are off and they metamorphose into this blob. So it's unknown whether these kind of snails actually were around during the Cretaceous period, parasitizing sea urchins and other kind of echinoderms. However, we'll probably never know because look at it, it's a squishy bag. These things ain't going to get preserved anytime soon. So far, I've been talking about like traces of parasites. However, it's a lot better to actually have the parasites themselves to actually study. And there is a medium that is particularly good for preserving parasites or evidence of parasitism, and that is coprolites. So coprolites are actually a very important and integral part of paleontology because while it's not looking through like what is essentially fossilized poop is not as glamorous as say putting together a dinosaur skeleton, fossilized poop offers so much in terms of what it has to tell us about the environment that the poop was left in or where the dropping was dropped, so to speak. So in these coprolites, you can find traces of like bits of leftover undigested material from prey animal, for example, if the dropping came from a carnivorous or omnivorous animal. You can also find bits of plant material. You can also find pollen, which can tell you something about the kind of flora that are found in the area. And of course, as I'm alluding to, you can also find eggs of parasites as well. Indeed, looking through the poops of animal and also humans as well is one of the standard procedure for figuring out what parasite someone or something is infected with, short of like killing them and cutting them open to look inside. So there is actually standardized techniques in both medical science as well as veterinary science to look through the fecal matter of various different kinds of animals to look for the eggs of parasites. And there is like a wide plethora of parasite eggs that you can find in poop. You can find roundworms and threadworms and pinworms. You can also find various tapeworms and flukes, not to mention all kinds of single cell parasites that have spores that can be found in the feces of their animals. So that's why you can find like a wide variety of them in the poop. The reason why so many parasite eggs end up in the fecal matter of host animal is that essentially because many of them live inside the gastrointestinal tract. So the gastrointestinal tract is like prime real estate for parasites. There's a lot of space for you to move as well as live in. There's also a lot of free nutrients in the form of pre-digested food. And also when you need to like produce eggs that complete the life cycle to go outside in the environment, all you have to do is lay the eggs and then the eggs will get carried out with fecal matter into the outside world. It's prime real estate. Furthermore, even parasites that live in other parts of the body, let's say liver flukes as well as blood flukes, they use various processes that I won't go into detail here to make sure that the eggs eventually find their way into the gastrointestinal tract so that they can be exported to the outside world in order for the parasites to start their life cycle over again. So it seems as if the anus of an animal is like the universal outbound port for all the parasites that are living inside of it. It's like, you got eggs, bring them down to the colon. We'll package them together in a nice chunk of feces and export them out into the outside world. So have we found parasite eggs in the coprolites? Well, thankfully we have. Otherwise, all that buildup would be extremely anticlimactic. So among other things, there's been tapeworm eggs that's been found in coprolites that's been attributed to sharks from the Permian period, dating back to about 270 million years ago. So tapeworms seems to have a very long history with sharks, even short of looking at the fossils. Judging from the molecular phylogeny, comparing the DNA, it seems that sharks were actually the original home of the tapeworm, the original host. So all of us who, like the rest of the animal kingdom that gets infected with tapeworms, they're all Johnny-come-lately. The sharks were the OG hosts for tapeworms. And it seems that they've been around for a very long time, since from the dating back from the Permian period and maybe even earlier than that, because there was a specimen of fossils of this particular shark that dates from the late Carboniferous period that have been found to have little objects that fits the size as well as the shape and the texture of tapeworm eggs associated with this species of sharks from the late Carboniferous period. These little eggs were actually found in what's known as colonite. So colonite is a subtype of coprolites. They're basically poop that hasn't technically become coprolites because they haven't been released into the outside world. It's still located in the hindgut. But in this colonite, it was found that there's these little egg-shaped, like parasite egg-shaped things that matches the size and descriptions of tapeworm eggs. So it's quite possible that tapeworm has an even older history with sharks. If anything, people kind of describe sharks as like living fossil, which I have issues with both the term and also sharks have actually changed quite a lot over all these millions of years. However, if sharks could be called living fossils, then I think shark tapeworms also deserve to be called living fossils as well. Now, there's also other kinds of animals, coprolites, that we have found parasites in. So for example, in various reptiles, including dinosaurs, if you're wondering, from the Mesozoic era, have also been found to have various different kinds of parasite eggs, including the eggs of flukes, as well as the eggs of nematode, parasitic roundworms. Now, the thing about parasitic roundworms is that different groups or different orders of these parasitic roundworms have differently shaped eggs. So judging from just by the shape of the eggs, you can kind of tell round about what taxonomic group that these particular roundworms belong to. And it seems that aside from flukes, these roundworm eggs belong to a group called the Ascarididea, which is today a very diverse group of parasitic roundworms. They have all kinds of life cycle, ranging from things like Ascaris, where the life cycle is very basic. Basically, eggs go out into the environment with the host feces, and then they stay in the environment for, it could be quite a while. If you accidentally ingest one of these microscopic eggs by not washing your hands and smearing it in your mouth, you will get infected. But there are also ascaridids with more complex life cycles. So for example, anisarchid nematodes that have a complex life cycle that goes from like a marine mammal into a crustacean and then into like a squid or a fish and then get eaten by a larger fish, would then get eaten by the marine mammal. So you have a wide variety of life cycle found in these particular group of parasitic roundworms, and it seems that they've been added since at least the Mesozoic era, since the Cretaceous period. And these kind of parasites eggs have also been found in earlier animals, for example, Parasites. So Parasites is the group of synapsid animal that include the ancestors of mammals. This is the group of animal that belong to a larger group called the synapsids that used to be a lot more diverse during the Permian period, except that most of them were wiped out by the end Permian mass extinction event. The only survivors were the ones that were the ancestors of mammals, essentially. And so these coprolites attributed to these particular animal from the Triassic period have also been found to contain various different kinds of parasitic roundworm eggs, including our friend Ascarididea, and also with pinworms as well. And finding pinworms in these particular coprolites is actually quite a big deal. Now, you might be thinking to yourself like, okay, what's the big deal with finding pinworm eggs inside an animal like this? All I know about pinworms is that those are the worms that make your butt itch, right? Well, the presence of the pinworm actually tells us a lot about the lifestyle of these particular animals. When you look at the fossil pinworm eggs, you can see that they look practically identical to like living pinworm eggs. I've included a photo of a living pinworm there for reference. But the significance of finding pinworms is that unlike other kinds of parasitic roundworms where they live inside the gut of the host and either feed on like host nutrients or maybe on like blood or other kind of host tissue, the thing that pinworms are really after are actually bacteria. And it just so happened that the hindgut of a variety of animal is filled with bacteria. So pinworms are found in a wide variety of animals today. They're found in many different orders of mammals, such as like rabbits, the lagomorphs, as well as rodents, as well as, of course, primates as well. They're also found in some birds, like ducks, like mostly herbivorous birds, and also in some arthropods even. So cockroaches and millipedes have pinworms living inside of them. The significance of this is that they are usually found in animals that have hindgut fermentation as part of the digestive process. So this is something that we probably won't be able to tell just by looking at even a complete skeleton of the host animal, because while you may be able to make inference about like, the size of their gastrointestinal tract and inference about like how big it is and stuff like that, this provides you with a bit of insight into the physiology of these particular animals that would not be offered by even the complete skeleton of the animal itself. So this is one of those reasons that I speak of why paleontologists should also pay attention to parasites, because parasites can offer some very key insight into the physiology as well as the ecology of their hosts. Usually pinworms are associated with hosts that includes a lot of cellulose or chitin in the diet. So this is a bit of insight into the possible diet of these animals that will not be offered short of like finding stomach content. And even then, it doesn't provide the level of insight as much as, say, some pinworm eggs in the coprolites would. Now, eggs in the coprolites of fossil animals can also provide other kind of insight about the paleo environment as well. So for example, these are echinophocephalan eggs. Echinophocephalans are also known as thorny-headed worms that's been found in coprolites that's been attributed to this Cretaceous crocodilomorphs called Ubarabasuchus. So these echinophocephalan eggs look practically identical to the eggs that are found in modern echinophocephalan. I've included a photo of an echinophocephalan here. There are many different varieties in terms of like shapes and sizes, but the common feature about them, indeed the common name, the thorny-headed worm, is named after this proboscis, which are covered in these hooks that allows them to cling onto the intestinal wall of the host. So they have independently evolved a lifestyle that is comparable to that of tapeworm, which is basically hanging around in the small intestine, absorbing pre-digested food nutrients through their tegument, through the body surface. Now, the significance of echinophocephalans is that with many of these parasites that have complex life cycle where they infect multiple different hosts in order to complete their life cycle, they follow certain rules of what particular type of host they have to infect. And in the case of echinophocephalan, when the egg goes out into the environment, the first thing they have to do is that they have to infect some kind of arthropod. So we're talking about, say, an insect or a crustacean. And then if that particular arthropod gets eaten by the final host, the life cycle is complete. In the case of some species, the arthropod might get eaten by another small animal, like small rodent or maybe a lizard or something like that. And then when the final host come along, let's say a predatory bird of prey or maybe a large reptile come along and eat them, that's how the life cycle is complete. So the presence of these echinophocephalan egg in the coprolites of these crocodilomorph tells us that either at some stage of their lives, these Ubarabasuchus ate arthropods, and or they ate things that ate arthropods. And they also provide us with insight into the paleo environment as well, because these particular worms would not exist in these crocodilomorphs if not for the presence of crustaceans and insects. and other arthropods. So just the existence alone can provide you with a lot of things to infer about the surrounding paleo environment. All of this is provided just by finding a few parasite eggs in coprolites. So you can see how insightful they can be to the study of paleontology and paleo environment as well. And you can do this with like a variety of other parasites as well. So for example, there's been parasitic eggs that's been found in coprolites from Spain, dating from the early Cretaceous period. It's not really clear what kind of animal produced those particular coprolites, but the most insightful thing about them is that these particular paleontologists found eggs of flukes inside of these coprolites. Now, the thing about flukes is that they follow a different set of rules to the Camphocephalon. The first host that have to infect is usually some kind of aquatic snail, because they also have an asexual stage in their life cycle. They go into the first host, and then they basically clone themselves and release a whole bunch of larvae that infects another host in the life cycle. So the majority of them use a snail, mostly aquatic snail, for this stage of their life cycle. There's a few families that use bivalves and a few freaky fish blood fluke that uses polychaete worm for this stage of the life cycle, but the vast majority of them, over 90% of all these digenine flukes, use snails for that part of the life cycle. So the fact that they even exist in the coprolite tells you that, oh, that particular paleo environment had aquatic snails in it. Even if you haven't found fossils of aquatic snails, you know that they will be there, because otherwise the fluke will not be able to exist in the first place. So that applies to a wide range of other parasite and like what influences you can make about the paleo environment through the existence of them and the kind of ecological interactions as well. Because as I mentioned before, many parasite transmission depend upon predator-prey interaction where one thing eats another. So the presence of these particular parasites can tell you a lot about what kind of other organisms are also found in the environment as well. Now, so far, all the examples I talked about are in coprolites, right? You have parasite eggs preserved in coprolites. And while coprolites is of interest to weird parasite nerds like myself and some paleontologists, there is also yet another material that is very good at preserving parasites and host parasites interactions in the fossil record. This particular material is coveted not just by scientists, but as well as members of the general public. And unfortunately, due to its popularity, extractions of it has also allegedly been involved in fueling armed conflicts in parts of the world, such as Myanmar. And that material that I'm going to be talking about is, of course, amber. So amber is this almost magical material when it comes to preserving biological and ecological interactions in the fossil record. There's been a wide variety of fossils been found of insects and other invertebrates, all kinds of ecological interactions occurring between them. So for example, mites that either hitchhike on the body of insects or even feed on them, so parasitic mites, as well as a wide range of other parasitoids that live in the body of insects and basically come bursting out of them like xenomorphs, and also a wide variety of other parasites as well. But the most spectacular example of parasite-host interaction found in amber is probably that of myrmidid nematodes. So myrmididae nematodes are this family of roundworms that infects. Sorry, I got interrupted by. I've just got a closed chat here. Sorry. Let me just close the chat window. Sorry about that. Can't go backward in this. Okay. So there's been fossils of parasitic myrmididae that have been found in amber. And what these particular parasitic nematodes do is that they get inside the body of insects and then develop to these alarmingly large sizes. And when they're ready to come out of the insect, xenomorph style, they alter the behavior of the insects so that they're more likely to end up in water, where the myrmididae can come out of the body of the insects and carry on with the life cycle. Now, the amber that has found preserved examples of this happening in situ date back from the mid-Cretaceous period. So they've been at this for a very long time, and they're not alone in doing this either. So for example, it seems that my slide has frozen. So, okay, all right, great. Just getting back into it. So there is another group of parasite that does pretty much exactly the same thing, which are the horsehair worm. So horsehair worms are nematomorphs. They belong, even though they resemble Mammiferae in just about every single way, including their life cycle, they are probably about as related to Mammiferae as sea squirts are related to us. They belong to an entirely different phylum that have independently evolved this way of life cycle where they infect an insects and then come bursting out of them at the end of their life, in hopefully in some water. And fossil records of these guys have been found from both the Eocene period, about 15 to 40 million years ago, to also the Cretaceous period. So around about the same time when Mammiferae was making life a living hell for insects, these horsehair worms are added as well. So there's been cockroaches in amber that's been found to have these worms coming out of their body. In addition to that, these nematomorphs are also well known from crickets as well as praying mantis as well. So you probably find videos of these worms coming out of the body of the host. It's actually quite alarming how big they could get. But these particular parasites are not the only ones that infect insects and manipulate the behavior. There's also the infamous cordyceps fungus. So there are a wide variety of parasitic fungus that infects insects, kills them, and then often bring the insect to places where they could sporulate better. And fossil record of these kinds of fungus date back from at least the Cretaceous period. And there are many different examples of this. The most well-known example is probably the zombie ant fungus. And much like what I said about the tongue biter lice, it's actually more accurate to call them zombie ant fungi because there's many different species of fungi infecting and targeting different species of ant, all causing the ant to do that thing where they climb up onto the underside of a leaf and then leave the fungi to sporulate. So you can find many different examples of this in the modern world. And they don't just target ants as well. They also target a wide variety of different insects. They even target like some other arthropods like spiders and millipedes. None of them are free from their influence. And it's really interesting that you start finding fossils of these things around about the mid-Cretaceous period, because what was happening during the mid-Cretaceous is what's known as the mid-Cretaceous terrestrial revolution. This was when the angiosperm, the flowering plant, started like really rapidly diversifying and flourishing across the landscape, which beget the evolution of many modern groups of insects. Many modern groups of insects got to be really successful, essentially riding on the backs of the success of flowering plants. And it seems that as those insects evolve, you also have these particular kind of parasitic fungus that follow these insects as well. So it's interesting to think that the diversification of flowering plants beget the diversification of many modern groups of insects, which in turn also beget the diversification of these particular group of fungi. And these fungi don't just leave fossils of themselves in a fossil record. They also leave possible traces. So for example, there are some leaf fossils dating from the Eocene period that have these really peculiar pinch marks on them. They have been interpreted to be what's known as ant death grip, because what happens when these fungus infect the ants and so-called zombify them is that when the fungus is ready to sporulate, the ant will leave the colony and then go climb somewhere relatively high and then clamp themselves onto the underside of a leaf. So many of the photos that you see of like, a fungus coming straight out the back of the ant, they've actually been turned upside down for aesthetic purpose. The fungus is actually dangling down from the corpse of an insect. So you can see there an ant kind of dangling, gripping onto the underside of a leaf and have this fungus dangling out of it. So you have evidence of this happening all the way from at least the Eocene about 40 million years ago, and it's quite likely that this particular type of fungi have been doing this for much, much longer. Now, there's also other amber preserve examples of parasites. So for example, there is a wide variety of blood-sucking insects as well that have been preserved in a fossil record dating from the Mesozoic era, mostly in the Cretaceous. Some of them actually carry passengers. So there are evidence that some of them might have carried the ancestors of the single-cell parasites that today causes, say, malaria, as well as leishmaniasis. So dinosaurs might have gotten malaria. We know that like living dinosaurs, i.e. birds, they definitely get malaria. So maybe that malaria has essentially followed the dinosaurian lineage since at least the Cretaceous period. But out of all the bloodsuckers, I think the one that probably really get under people's skin and causes many people to shiver is probably the ticks. So ticks, judging from the DNA comparisons, seems to have had a relatively long evolutionary history. Based on comparisons of the DNA of different groups, it's been estimated that they originated during the Carboniferous period. This is also a period when many vertebrate animals become established on land. So as soon as vertebrate animals started making themselves at home on land, it seems that the ticks were around the corner going, yes, ha ha ha, yes. And also, one of the main division within the ticks is between the so-called soft ticks and the hard tick. And based on comparisons of the DNA, this might have originated during the Permian period, which is also when you have the big split between the so-called amniote groups of animals. So amniote animals are the group of terrestrial vertebrate animals that don't need to go into water to breed. So you have the two main groups. You have the synapsids, which is the group that contains the mammals, and also the sauropsids, which are the group that contains the reptiles as well as birds. And it's interesting that that particular split happened close to around about the time period when this major split in tick evolutionary lineages also happened as well, between the hard tick and the soft tick. So what about the actual fossil records of ticks? Well, unfortunately, the fossil record of ticks only stretch back to the Cretaceous period, mainly because amber fossil only become common round about from the Cretaceous period onward. There are amber dating back all the way to the Carboniferous period, but they are relatively rare. Like, it wasn't until the Cretaceous period where you start finding a lot of amber fossil, because the plants that produce the kind of sap and resin that can be preserved as amber weren't really, really flourishing and abundant until the Cretaceous period. So there are fossils of ticks dating back from the Cretaceous period, and they've been associated with feathers of what has been purported to be non-avian dinosaurs. So yes, dinosaurs had ticks, and not just any ticks, but these ticks that seems to belong to a group that is no longer alive today. So these ticks probably died out with the dinosaurs at the end of the Cretaceous period. There's also other kinds of ticks that's been found that's really intriguing from this particular time period, including this tick here that I'm pointing to with a red arrow. The interesting thing about this tick is that it actually combined features of both the soft ticks as well as the hard tick as well. So there might have been a completely extinct lineage of ticks that have existed back in the Cretaceous period, and sometime between now and then have gone extinct. So that really adds to our understanding of the family tree of ticks, I guess, if you want to put it that way. Now, this kind of raises another question. Given that I mentioned that dinosaurs probably had tick, that raised the question of what kind of parasites are found on dinosaurs, which I'm sure there is a significant number of the members of the audience that are dying for me to answer this question, which is what kind of parasites are found on dinosaurs? We've watched that episode of Prehistoric Planet where a dino carrier scratches itself. What was it scratching itself against? Well, let's have a look at the living dinosaurs for comparison and some reference as to what might have bugged the dinosaurs, so to speak. So living dinosaurs, i.e. birds, have of course ticks. They also have mites that, depending on the mite, some of them live on feeding on just detritus like skin flakes as well as bits of feathers. Some of them are actually bloodsuckers that feed on the blood of birds in a comparable way to ticks do. There is also this particular group of flies called louse flies, which are essentially a group of flies that have evolved to be specifically ectoparasites of warm-blooded animals. They have these really sturdy legs that allows them to scurry really quickly, disconcertingly quickly across the body of the host to avoid being groomed off. But by far, the two groups of insects that are most commonly found on birds are the Siphonophorans, which are the fleas, and the Pteropterans, which are the lice. Lice are extremely common on birds and also very diverse as well, because each lice tend to be very specialized on what bird that they live on. So the fossil evidence for recognizable fleas and lice only date back from the Eocene period, around about 40 million years ago. So it seems that there are no fleas or lice dating back from earlier, from when the non-avian dinosaurs lived. Or is there? So Right about 10 years ago, there was a Nature paper that was published by some researchers claiming that they found these so-called giant Jurassic fleas. It was received with like a great deal of fanfare and sensationalism because like it really captured people's imagination. Like, wow, giant dinosaur fleas from the Cretaceous period, from the Jurassic period, like just like in the movie Jurassic Park, right? However, there are some researchers, including parasitologists and specifically entomologists who study fleas that have some criticism or questions about this particular interpretation of these Mesozoic insects. First of all, unlike many of the parasites I've talked about previously, these so-called giant Jurassic fleas, giant dinosaur fleas, weren't actually found in association with any host material, which is very unusual for parasites. Most of the time when you find parasites, they are found in association with like at least some host material, bits of feathers or bits of other host material. Additionally, there's no dinosaurs that's been found in same assemblage, like in the same area as these so-called dinosaur fleas. So the researchers basically assume that like, oh, they're big bugs from the Mesozoic and they have biting mouthparts. They must be fleas of dinosaurs. The actual fleas expert, researchers, entomologists, and people who specifically study fleas also point out that the morphology, the anatomical characteristics of these so-called giant Jurassic fleas are not homologous with actual fleas. Now, a little bit about homology. Homology is where, for example, if you look at your forearm versus that of, say, a whale's limb or an armadillo's claw or a bat's wing, they're all very, very different. However, if you look at the individual bones that make up those structures, you'll find similarities because we share a common ancestry. What these flea experts are essentially saying is that these so-called giant Jurassic fleas do not share any of the similar characteristics with fleas, and they're different from fleas in a way that indicates that they don't share a common ancestry. So to them, they're definitely not ancestors or related to fleas in any way. Another key thing too, and this is like a very common thing that seems to pop up, is people's assumption about characteristics or structures that are adaptations for parasitism. So the researchers who published that Nature paper, they claim two main features for interpreting these bugs as dinosaur parasites. First of all is piercing mouthpart. Now, piercing mouthpart is not an exclusively parasitic features. There are a wide variety of insects out there that are not parasites at all that have piercing mouthparts. So, for example, many predatory insects like giant water bugs, assassin bugs, robber flies, they all have like piercing mouthpart for stabbing into the prey and sucking out juices. The other feature that they cite as characteristics for parasitism is tarsal claws. So tarsal claws are the little claws that you find at the end of the legs of insects and other small arthropods as well, small terrestrial arthropods as well. But the thing is that tarsal claws of actual parasitic insects, like for example, lice and fleas, are often, and also those like louse flies I talked about, often a lot larger and curved, which allows them to grip around shafts of, say, feathers or bristles or hair or whatever other material on the surface of the host. The tarsal claws of these so-called giant Jurassic fleas are relatively small. In fact, they're probably around about the same size as you would expect from regular insects like beetles and crickets and cockroaches, because it just so happened to be useful for a small animal to have claws that allow them to climb up something. And it's not necessarily the flank of Brontosaurus or something they're climbing up. So this seems to be a reoccurring problem in the literature. When I read through the literature, I find many accounts of this, where you have multiple examples of just literally random Mesozoic fossil insects that get interpreted as dinosaur fleas, because it's more exciting that way. And many of the interpretation are based on what I would call just so stories. Like, for example, piercing mouthpart must have been used to pierce the flank of a dinosaur, as opposed to any other explanation. They often don't offer other potential and equally viable explanation for the lifestyle of these rather interesting looking Mesozoic insects. And it seems that the idea of dinosaur flea is more infectious than the actual insects that are being described. And I know that this particular meme that I posted up here is rather a bit flippant. However, it would be flippant if it isn't a problem that keep on happening in the literature when I read it. So, for example, this peculiar insect is called the Strassilidae. When it was initially discovered and described, it was described as a pterosaur ectoparasite. For the usual reason, piercing mouthpart, and it was interpreted that those back legs would have been used to clamp onto the hairy pycnal fiber on the surface of a pterosaur. Now, the issue with this is that when I, as a parasitologist, look at those hind legs, they don't look like they're good for clamping around hair or any other kind of material like that. As I mentioned before, when you look at actual parasitic insects or ectoparasitic insects, they often have features that allow them to wrap around a bristle or hair shaft or something like that. But there's no such feature on the legs of the Strassilidae. If anything, they look more like legs of like a cricket. They're not very good at like gripping around things, but they are very prominent legs. And later on, it was discovered in another set of fossils that they're not really parasitic at all. In fact, these ones with the massive legs are probably males of a species of amphibious flies. So these fossils that have been found to have these, in mating position, and also those little strands of things coming out of the abdomen are actually gills. And so it seems that Strassilidae isn't parasitic at all, has nothing to do with parasites or parasitism. It's just some random fly that happens to have some features that one particular paleontologist really caught on and go like, these must be parasite. Another example of this is with this guy, Saurophyropsis or Saurophysis. They have also been interpreted as one of these like giant Jurassic fleas on the backs of, basically piercing mouthpart. The legs aren't really well suited for ectoparasitism, as you can see from the last slide that I talked about before. They might have long legs, but they're usually like really stout. They don't miss leg day because it's useful for scrambling over the surface of like a mobile host, but that's not the case for Saurophysis. Nevertheless, the meme of these guys being parasite is so common that it actually made its way into an episode of Walking with Dinosaur, where one of these bugs was seen feeding on the wing membrane of a giant pterosaur. Now, aside from basically anatomical features of these Saurophysis not supporting the idea of them being ectoparasites at all, there's actually evidence against these guys being ectoparasites. And they were found by none other than two researchers who were looking for evidence of parasitism in these insects. So when they look at the gut structure of these insects, they found that it doesn't really match that of a blood-sucking insects. And additionally, when they did element analysis on the gut content, they didn't find what you would expect to find in blood-sucking insects. Usually in blood-sucking insects, you would find a lot of iron as well as traces of organic material because, that's what blood is composed of. In fact, there are early Cretaceous insects we can turn to look for like comparable examples of this. So these particular bugs are related to things like kissing bugs and bed bugs, which are hemiprons that are blood feeders. Under element analysis, it was found that especially in specimens that have recently fed, the gut as well as the mouth part is full of iron. But even in specimens that haven't recently been fed, there are high level of iron in various different parts of the body comparing with the background material because blood feeding insects also incorporate that iron into various different parts of the body as well. This was not found to be the case for Saurophysis. So in the light of this particular finding, the researchers changed their mind and reevaluate their assumptions of Saurophysis. It's what I would have liked to have said, because if you read the discussion of the paper, they essentially doubled down on the idea of these guys being pterosaur parasite. They were so in love with the idea of these guys being pterosaur parasites, they came up with all kinds of convoluted reasons for why there isn't actually any evidence for them being parasitic. In fact, there are evidence against them being parasitic. And I kind of wish when I read these studies that any one of these paleontologists would actually like speak to the local entomologists for once, which would be able to tell them like, yeah, this ain't it, chief. Because you have things like this being published. So for example, this is a more recent example, Misinterpreted, which was described as a kind of a dinosaur lice. At least I'll give it that they actually find them on some feathers. So at least they have like that reason to believe that it was a dinosaur lice. It has like this nice reconstruction of these cute little bugs on the feathers of dinosaurs, except that it's not actually accurate because this paper was read by David Grimaldi, who wrote a massive book on fossil insect, as well as Isabelle Viel, who is an expert on scale insect, which are these tiny little sap-sucking insects. And they take one look at these bugs and they go, yeah, they're not dinosaur lice, they're scale insect. You know why? Here's an example of a scale insect from the modern day. And look, they look practically identical. I mean, it's pretty amazing that these particular insects haven't changed for like 100 million years, but they're not parasites of dinosaurs. And there's plenty of reasons to that they would have been preserved with like a bit of loose feather or from, say, a passing dinosaur. So it seems like a lot of this could be avoided if there's like more interdisciplinary research between, say, paleontologists and entomologists and parasitologists, people from relevant fields, because it seems that many of these reconstructions of these random Mesozoic bugs living as dinosaur parasites is about as accurate and about as out of date as that of those bizarro reconstructions of Quetzalcoatlus as this demonic vulture of the Cretaceous. So I'm sure some of you will be thinking like, hey, you know, speculation is like a big part of paleontology. You know, like science is a self-correcting process. So as long as like eventually things get corrected, it'll be okay, right? Well, the thing that frustrates me about all these random bugs, like fascinating looking bugs, but like random bugs getting described as dinosaur or pterosaur parasites, is that it's what's known in sport as an unforced error. Like we could have avoided like putting in a lot of these misleading information into literature if any of these paleontologists have actually spoken to like a parasitologist or an entomologist. It also muddles like entomology as a field. Like if you have people who actually want to study the evolutionary history of like fleas or scale insects and stuff like that, it doesn't help that you have these bunch of random overzealous paleontologists who are like describing these things as like dinosaur fleas or dinosaur lice when they're actually not. And as far as we could tell, recognizable examples of fleas and lice only date back to about 40 million years ago during the Eocene period. And trying to understand the evolution of these particular insects are not helped by having paleontologists describing random things as dinosaur fleas or dinosaur parasites. Now, I understand that at this point, I'm sounding like a bit of a hater. So I'm going to try and put a positive spin on this particular phenomena. The fact that you have a bunch of overzealous paleontologists out there describing every random Mesozoic bug they find as a dinosaur parasite tells me that there's actually a lot of interest in fossil parasites. And I see this as a great opportunity for potential interdisciplinary collaboration between these different fields. So, you know, I recognize that paleontologists have some very specialized skills that are fantastic for finding fossils, as well as extracting them and preparing them and interpreting them. However, in the hunt for fossil parasites, if they're serious about this, this could be greatly complemented by the skills and know-how of entomologists and parasitologists who are very familiar with the morphology of actual parasites and know what to look for. So for this, I guess, final part of the talk, I'm going to provide some kind of theoretical framework for going forward in terms of research directions and where we could go with fossil parasites. Because just to start with, the fact that dinosaur does not appear to have lice and fleas provides us with some really interesting research questions. For example, did insects live at all as ectoparasites of warm-blooded terrestrial vertebrate prior to the end Cretaceous mass extinction event? We know that at least some, like for example, those hemipteran bugs, would have fed on the blood of vertebrate animals. However, were there other insects that were also living as like ectoparasites, if not for, you know, fleas and lice, which didn't evolve until afterwards? And were these insects actually the actual ancestors or predecessors of modern fleas and lice? And if not, then what were the dominant ectoparasites of dinosaurs and also mammals and other kind of warm-blooded animal during this particular period of time? So part of, I guess, the hunt for fossil parasites and understanding where to find them and what they might look like is to understand the framework under which they might exist. And I'm going to be providing this framework in terms of trying to anticipate what you might find in a fossil record. And I'm going to do that by borrowing a technique from paleontology called extent phylogenetic bracketing. So extent phylogenetic bracketing is a technique for basically trying to guess at the trait of extinct taxa. or extinct groups of animals. So for example, this is Anchiornis. Anchiornis is an extinct theropod dinosaur. And let's say the question is, did it have a four-chambered heart? Now we know that crocodiles have four-chambered heart and birds have four-chambered heart. And given that Anchiornis is nested within the group that contains both crocodiles and birds, it's pretty safe for us to assume that these dinosaurs probably have four-chambered heart as well. And you can also apply this to parasites and parasite communities as well, in terms of trying to guess what kind of parasites are found on what particular kind of host. So for example, this question that I proposed, like what vertebrate animals were infected with tapeworms, specifically adult tapeworms living in their gut. Now, among the living groups of vertebrate animals, basically tapeworms are found in like everyone. Everyone has tapeworm, ranging from lamprey to cartilaginous fish, which, like I said, is the original OG home of the tapeworm, to rayfin fishes, as well as the Sarcopterygian, the lobe-fin fish, which includes ourselves. So all of us have tapeworms. What about all of these extinct groups like the placoderms and the phelodonts and the acanthodonts and stuff like that? We're not sure whether they had tapeworms because we haven't found fossils or fossil evidence of tapeworm from these particular animals. However, by knowing that there are tapeworms found in all the other groups of vertebrate animals, it's a good possibility that those guys, these armored fishes, as well as rayfin fish and even some of the jawless fish, might have had tapeworms as well. Now, this particular framework is a good place to start. It does have some caveats though, because we have to also take into account whether the fossil record for tapeworms stretch back that far. Given that the oldest known tapeworm fossil eggs look pretty much like what you expect tapeworms to look like, it's quite likely that they actually have a fossil record that date further back than, say, the Permian or the Carboniferous period, but we don't know at this point. But at least this is somewhere to start. It provides paleontologists with something to look out for when it comes to what kind of parasites are found in this study taxa. Now, there are also other extinct taxa which are not really all that comparable to the closest living relatives. So for example, Dimetrodon is one of these like synapsids, proto-mammals that, as you can see, look very different from their closest living relatives, which are mammals essentially. You also have things like large theropod dinosaurs, as well as sauropod dinosaurs, which are very different from their closest living relatives, i.e. the birds. And also with certain groups like Sauropterygians, these like marine reptiles from the Mesozoic era, which don't have any living relatives at all. So in the case of animals like this, where we don't have living relatives to compare to figure out what kind of parasites might have lived on them, I propose something called ecological fitting. And this is a concept that's broadly comparable to convergent evolution. So convergent evolution is where you have different organisms living the same in same or similar habitats or lifestyle, evolving to have similar body shapes or morphology. The textbook or even the kids' book example of this is dolphins versus ichthyosaur. Both of them lived as pelagic predators, and so large pelagic predators, there's some evidence to even indicate that ichthyosaur might have been, at least to some extent, warm-blooded, and so they have evolved a similar kind of body plan. And this could also apply to the parasites as well. When you look at actual living animals, even very different animals often have similar parasite communities living inside of them because they have similar ecological niches. So one example of parasite being shared between animals that are taxonomically very different is with these guys, these acanthocephalans. This species is called Carina australe, and they're mostly found in sea lions and seals. However, they're also found in another marine animal, Magellanic penguins. So the common ancestor of these two animals date back to like about 300 million years ago. However, because there's enough similarity in the physiology and most importantly, their basically their ecological niche in terms of what they eat, they all end up sharing the same parasites because the sea lions get this particular worm from eating fishes, and penguins presumably also get this worm from eating fishes. So they've been eating the same kind of fish. And so the body of of these penguins are similar enough to that of sea lion for these parasites to thrive. It's like, well, this is not what I expected, but it all kind of worked out better than I thought. As an additional twist to this, these particular parasites are actually originally found in seabirds like albatrosses and the like. So at some stage during the evolution of these worms, they made the jump to sea lions and then made the jump back to birds, but a very different groups of birds, so to speak. So it's quite possible that these kind of processes have occurred during the geological past with various different groups of animals as well. And this is even more striking when you look at, say, parasitic flukes in whales, because whales have a wide variety of flukes living inside of them. But in contrast to what you might think, the flukes that are found in whales, the closest living relatives aren't actually found in other mammals. They're found in other kinds of animals like birds, as well as fishes. So the photo that I have here is of Nasty tremor. Nasty tremor is a type of fluke that lives in the cranial canals of whales. For a while, people believe that they're associated with like causing whale strandings because they live near the brain, and often stranded whales are found with these flukes. But it's most likely that the reason why stranded whales have these flukes is because they're whales and they have these flukes, and it's got nothing to do with their presence that caused the stranding. However, when you look at the evolutionary lineage of this particular type of fluke, you'll find that the closest living relatives are actually in fish. So during some stage of the evolutionary history in the geological past, the ancestors of these flukes made the jump from living in a fish host to a mammal host, which it seems like an insurmountable gulf given the differences in the physiology as well as the anatomy, but somehow they managed to make it. So it's possible that the same process has occurred with many of these marine reptiles in the past, and that they might have shared some of the parasites that are found in modern marine mammal, might have had relatives that were actually found in marine reptiles as well. Or maybe they went through an independent set of host jumps that resulted in the parasite communities. So by understanding how parasite communities are formed in living animals, that gives us some insight as to what we might want to look for in extinct animals in their fossils as well. So in conclusion, fossil parasites can provide some key information about their host as well as the paleo environment. So that's the reason why paleontologists should really get interested in fossil parasites. And many modern groups of parasites actually have recognizable fossil relatives dating back from the Mesozoic era or even earlier than that. Parasite communities, of course, of modern host species can also inform the search for fossil parasites. If you know what kind of parasites are generally found in animals that are comparable to your particular study taxa, that provides us with some clues as to like, okay, what we should be looking out for in terms of parasite material in the fossil record for your particular study taxa. And also the search for fossil parasites also present many opportunities for potential interdisciplinary collaborative research. Like, I don't know about other parasitologists, but I'm super keen to get on board with like studying more about fossil parasites. So if there's paleontologists out there who are interested in looking for parasites in the fossil record, hit me up sometime. So that's pretty much all I have to say about fossil parasites. Thank you for watching and listening, and sorry about the little bit of technical interruption early on. I'm going to leave you with this image, which is of Eric, the opalized plesiosaur from the Australian Museum, and also this ecophycephalon, which is a giant ecophycephalon found in the guts of pigs. So, any questions, I guess? I see that there's already a question in the Q&A about parasite-induced behavioral change. Yeah, so in terms of where they're found inside of the host, so the Myrmifidae nematodes and the nematomorph, we know with modern examples that they can be found, they often alter the behavior of the insect host in such a way so that they're more likely to end up in water. And given that's what they do today, the closest thing we have is inference that like they probably did this in the past as well. So it's difficult to get like direct evidence because they're fossils, you know, you don't see them behaving in any way. So it's difficult to infer behavior. However, judging from what they do to the host today, it's quite likely that they probably did that during the Cretaceous period over 100 million years ago. So aside from the chat question, I'm sure you have other people in the audience that might want to ask a question as well. I'll leave it to you.
All right, thanks, Tommy. That was a very thorough and interesting talk.